Hello everybody. Good evening and welcome to the extra course or extra series lectures for this course, Classics in Neuroscience, that you have opted in your NPTEL. So what we'll be doing here is we'll be going through the previous year questions for week 4 particularly, and we'll see if going through the concepts behind answering those questions actually help you in solving the current year assignment as well. Along with that, what we'll be doing is we'll be discussing in detail certain concepts that have been mentioned during Sir's class, including the in-depth review of the neural circuit as an neural network as an equivalent circuit, wherein we can understand the GHK equation, Nernst equation, the conductances and impedances better. Also, from ahead of that, we are also going to understand how these channels are working in and how they are contributing in basically determining how an impulse is created or how an action potential is finally determined. So, we have already looked at where the action potential is generated, how it is being conducted in the previous session. Today, we are going to understand a little more about the molecular mechanisms of which lead to the action potential and how those mechanisms have actually been deduced, like which channels are involved, how do we know that what is the involvement of a particular channel. So all that we are going to understand. So that is going to be the second part of the lecture. For now, we'll begin with the previous year questions. For week 4 lectures, there were a few that were repeated also from last week's week 3 lecture questions, for instance, this one, and it was fairly simpler, right? So, as you go ahead in the course and you build a better understanding, you will also feel that the questions are getting simpler to answer, right? For instance, this one, which is who coined the term synapse and helped describe its functions. So the options that were given were Ramonica Hall, Bernstein, Sherrington, or Eccles, right? So just a moment, yeah, yeah. So the correct answer for this one is Sherrington, right? Charles Sherrington. He was the one who coined this term synapse, and it is basically a term that is used to describe the region or the junction between the presynaptic and postsynaptic neurons, wherein the transfer of information is actually happening, right? So that portion is known as synapse. Then the next question was, what determines the frequency of miniature end plate potential or MEPP? Right? So is it the presynaptic terminal or postsynaptic terminal or calcium levels or membrane potential? Right? So if we understand just what miniature end plate potential is, it is very simple for us to answer this question, right? So the correct answer over here is presynaptic terminal. Why? Because the amount of neurotransmitter that is going to be released from the presynaptic terminal is what is going to determine how much potential is going to be generated at this basically interface, right? So, for instance, I have kept this representation from Candle, uh, the book by Candle, right? And over here you can see that the neurotransmitter is released in fixed increments, right? So some a little bit is given, then more of it is given, then more of it is given. So there are fixed increments that are done. Each increment or quantum of transmitter produces a unit excitatory postsynaptic potential of fixed amplitude, right? The amplitude of EPSP evoked by nerve stimulation is thus equal to the amplitude of the unit EPSP multiplied by the number of quanta of transmitter released, right? So the end plate potential, what it means is, or what we want to understand by this is that the end plate potential, whatever end plate potential is getting produced, is heavily dependent on what is the amount of neurotransmitter that is getting released from the presynaptic terminal, right? Because that over here is almost like a sole determinant, right? There are other factors such as the receptors should be there on this side of the terminal, etc. But this is a major determinant in that, right? So that is why over here, for instance, you can see the amplitude of spontaneous miniature end plate potentials in millivolts that is plotted over here, right? Yeah. So moving forward, the next question, again, a very simple one is why was the aplysia considered as an important model system for neuroscience, right? Uh, A, because it had a complex brain. B, because it was found in many countries. C, because it had a simple nervous system. Or D, it was very large in size, right? So, which one of this would be the correct answer? So this is aplysia, right? And the correct answer is because it has a simple central nervous system. It is not too complex. So, if you are basically trying to stimulate some region, you can directly observe the response that it generates within the nervous system, right? So you don't have to delve very deep. There are not too many processes that are involved over here. So relatively, it is simpler. So that is why it is a good model organism. Then, what type of recording method did early scientists like Edgar Adrian or Zoterman use to study sensory receptors, right? Was it intracellular recordings or extracellular recordings or brain imaging or microscopic observations? So, the hint for this answer is that they are saying that early scientists used this method, right? So it was a less sophisticated method, most probably, right? Brain imaging came much, much later. It is a part of contemporary neuroscience. We have not even started to understand that part as yet, right? So contemporary neuroscience we have not reached. So brain imaging is in that era where CT, MRI, etc. have come into picture, but that has not happened during this the era of these early scientists, Adrian and Zoterman. Microscopic observations were have been around for a while, but it is not a recording method, right? It is a histological method, so we can rule this one also out. Then remains intracellular recordings and extracellular recordings. Now, because it is saying early scientists, so it would be a, as I said, less sophisticated technique than what we have today. So you can safely assume that they are talking about extracellular recordings, right? So intracellular recordings means recording from a single neuron. Extracellular recording means the electrode is near a bunch of neurons, so the input that it is getting or the reading that it is obtaining is an a multi a summated response of a multitude of those neurons that are present around it, right? So it is a summated response because it is in the close vicinity of a lot of neurons. It is not taking the recording from a particular neuron, right? So, for instance, over here you can see uh, this is an example of a normal muscle versus a paralytic muscle, right? So, uh, again, another representation that I have picked up from Candle's book. So over here, you can see this is a neuromuscular junction that is represented over here. This is a neuron. This is the muscle spindle or muscle fibers that are present, and they have shown that this muscle fiber, it has a lot of sodium channels on its surface, right? So this one, let's discuss about the A part of the figure. So the electrical signature of myotonia. What is myotonia? It is muscle stiffness, right? Or even spasming of muscles sometimes is referred to as myotonic, right? So, it is a rapid burst of action potentials in response to a single stimulus, right? So that is why spasming occurs. What is spasmic? Like it is a contracted state which is not voluntary, right? There is involuntary contraction sometimes that occurs. So, the action potentials shown here from the extracellular recordings vary in amplitude and wax and wane in frequency. Such a burst may be triggered by voluntary muscle contraction or mechanical stimulus such as percussion of the muscle. Right? So, why are we not seeing a single peak and why are we seeing a burst of peaks? It is also because it is an extracellular recording, right? Or what did I just do? Let me reshare the screen. Yeah. Then this part, if we talk about, right, it is a cell patch recording, right? So we are just simultaneously, once we have all gone through extracellular recording, let us understand a little bit more about the recording methods, right? And what is the output of these methods. So when we talk about cell patch recordings from a cultured human muscle cell, so what was done over here? The human muscle cells were isolated from maybe muscle biopsy or cadaver muscle, which is relatively fresh, and these muscle cells were isolated and they were cultured for a while, and then the cell patched recordings were done. That is basically single cell recording of these cells, right? In normal muscle, the sodium channel open early and briefly in response to a voltage clamp depolarization from minus 120 millivolt to minus 40 millivolt, right? So this means that we know that muscle, like the resting membrane potential is more negative when it, I have told you, when it starts to move towards zero, that is known as depolarization. That is when the action potential is also attained and so on, right? So over here they are talking about a similar event wherein they are saying that what they are doing is it is a voltage clamp, meaning you can control the voltage. So what they are doing, they are changing the voltage, which was earlier it was 120 as you can see over here, and then they are suddenly changing it into minus 40. So what happens in a normal muscle is that the NA channels open, but they open briefly, right? But in case of a paralytic muscle, what happens? Uh, Patients with hyperkalemic periodic paralysis, prolonged openings and reopenings indicate impaired inactivation. So what this means is the sodium channel has opened over here, but it continues to remain open for so long, right? This means that we talk about an inactivation phase. When I was last week when I talked about how the synaptic transmission is happening, not the synaptic, axonal transmission of signal is happening, where it is getting generated at the hillock and it is moving forward. Why it can't, after a point, why it can't move backward is because the receptors over here will be in an inactive phase. So there is just one direction. So it is pushed forward. That a portion that we talked about is how there is efficient transmission that can happen. But over here what is happening is the inactivation part is not working nicely, right? So it is opening but it is not closing. Once something happens, there should also be a reversal of that so that balance can be attained, right? So that balance is not getting attained over here, due to which this kind of paralysis is actually getting caused. So the probability of channel opening, which is obtained by averaging individual records, remains elevated in the hyperkalemic muscle following inactivation. So after inactivation, ideally for a transient period of time, there should be no other activation that is possible. But over here, that is not the case, right? So that is why probably this kind of an effect is being caused, paralytic kind of effect, right? So even modest disruption of sodium channel inactivation is sufficient to produce bursts of myotonic discharges or depolarization induced loss of excitability, right? These computer simulation records show muscle voltage in the response to an injected depolarizing current. So they are talking about this now, right? Oh no, this one. Um, where were we? The fraction of mutant channels that actually fails to inactivate normal varies within a population over time. In these simulations, F was varied from normal to values appropriate for myotonic or paralytic muscle. So it is a simulation that is shown over here, right? This one. So I hope if at least you have attended the previous session and the latter part of the previous session wherein I was talking about this action potential generation. At that time, we have briefly talked about why inactivation is important, right? I will again talk about it when we discuss how exactly those action potentials are generated in today's session also later on. So then maybe you'll get a better understanding or clarity of this part. Then what is recorded from frog olfactory epithelium in response to smell stimuli, serving as a useful monitor of overall stimulating effectiveness of different odor compounds. Is it electroretinogram ERG or electrocardiogram ECG or electroolfactogram EOG or electroencephalogram or EEG, right? So again, the question has the answer within itself. Olfactory epithelium is being recorded, so it is going to be electroolfactogram, right? Also, EOG stands for electrooculogram as well, right? So retinogram is for the retina part of the eye. Oculogram is for the is also used for eye based recordings also when it is not particularly for retina. Okay, so how does information first enter a neuron, right? So I think this is a very basic of neuroscience, neurobiology, right? So you will know this by now that does it enter through an axon or the cell body or the dendrites or the nucleus, right? So what is that portion you can see in the background over here also, which is going to come into contact with the other neurons, right? So it is going to be the dendrites first, right? Over here, the dendrites. So this is a representation of the neurons that are there in the spinal cord, for instance. And you can see that the dendrite of this neuron is going to come into contact with the C terminal or the axon terminal of this other neuron. So this is going to be the first region of a neuron through which information will enter this neuron, right? So, what technological advancement helped support the neuron doctrine in the 1950s? Was it the computer technology which was advanced? Was it the electron microscope that was introduced, or the X-ray machines, or the telescope? Right, so definitely we don't need a telescope when we are looking into cells and into the brain, right? That is for extra celestial objects. X-ray machines, yes, it came into picture, but it came into picture a little later than 1950s when we are talking in context of brains. So CT was developed based on the principle of X-ray, for instance. Computer technology also a little later it developed, computational part of neuroscience, but electron microscope was the one that was developed around this time, and it also helped support the neuron doctrine, right? Because now through electron microscope what we could do is we could look at the ultrastructure of the brain, and we could see that yes, indeed, neurons are not, it is not one continuous network of neurons. So reticular theory was definitely disproved, and it was definitely identified that yes, there are separate neurons, they have gaps that exist between them, right? And this was finally proved. So then the next question is, where did researchers discover action potentials are most likely to be generated in a neuron? Right, so again, this is something we have already discussed last time. Was it at the axon hillock, or near the nucleus, or in the dendrites, or in the cell membrane? Right, so it is most likely to be generated in the axon hillock. Again, this is the representation that I have picked up from the previous discussion that we had last time, right? So this is the nucleus, this is the cell body of the neuron, this is the axon, this is the axonal terminals, right? And the almost the beginning part of the axon is where the axon hillock is, right? And when I discussed this, I also told you why generally the potentials are generated here. It is because of two reasons. One is because there is a high density of these channels over here, so sodium potassium channels, right? So the exchange can occur in that bulk amount so that it can generate or cross that threshold. Also, the threshold that has to be crossed is lower over here as compared to, say, in dendrites or cell body, right? So these two things, they favor one another, and it is generally the region where the action potentials are generated in a neuron. Then question number 9 is, what was a key discovery about dendrites that challenged the original understanding? Was it that the dendrites are completely inactive, or that the dendrites can only receive the signal, or the dendrites can also conduct action potential and receive feedback, or the dendrites are only found in specific type of neurons, right? So no dendrites are found in all types of neurons. They are not just receivers of signal, although that is majority of the part of work that they do. And they are also not completely inactive, because even in receiving signals, they are working, right? So option C is the correct one, that they can also conduct action potentials and they can receive feedback. So for instance, through dendrodendritic neurons, right? So that is feedback mechanism that is happening. Then what unique term did H.T. Chang introduce to describe dendritic spines? So he was one of the people who helped come to this conclusion that they are also not inactive. They can also receive signals. They can also generate potentials, those things, right? So which of these terms he used to describe these dendritic spines? Was it neurons, or gemmules, or synapses, or cables? So the correct answer for this is gemmules, right? So he also called them as gemmules. So that is the end of the previous year question part, right? And we can move on to our discussion for this week. So, for people who have joined a little late, I would like to reiterate what we'll be doing over here. So, we'll be talking about some of the topics that have been discussed by Sir also, or have been spoken about by Sir in his lecture as well. We'll have a more detailed discussion about that. The major part of the discussion will account for two things. One is that we'll discuss the equivalent circuit of the neural unit as an equivalent circuit, so that we can understand how the resistance, capacitance, impedance, etc. come into play in case of a neuronal circuit or a neuron. We will also look at, and this will eventually help us derive the GHK equation and the Nernst equation as well. Also, another thing that we'll be looking at is how an action potential is actually developed, right? So how it is getting generated, what is the cause of the action potential getting generated, which channels are responsible for that, how that study has come into picture, how it has been reduced, that part also we are going to look at, right? So, before we begin this part, please allow me like 30-second, 1 minute break. I'll just be back. Thank you for the wait. I'll just reshare my screen. Okay, so we can begin with this part. So we are going to begin with the discussion of a few. Before the scientists, we are actually discussing the reflex action, right? Because Sir has also briefly mentioned this, so I thought I can keep this. It might be useful for a lot of you to understand certain responses. So the first thing we are going to discuss about is the sequence of signals that produces a reflex action. I am assuming that everybody over here is aware about what a reflex action is. Still, in one line, we would state it again. So, it is basically an action that is produced in a fraction of a second, wherein the response directly happens with the communication through the spinal cord, and this information that generates or calls for this response, it doesn't get transferred to the brain itself. So because it is probably a situation that requires a quick action, right? So that is why this quick action, it is modulated through the spinal cord neurons itself, right? So that is why it is a very quick response, and it is known as a reflex action. For instance, we have when we were younger, we used to study these instances wherein if you touch your finger to a vessel that has a very that is very hot, you immediately remove it. So where is that information coming from? It is coming directly from the spinal cord, right? Because if that information was taken to the brain, it was processed. Although our brain processes things very fast, right? I am speaking continuously, you are hearing what I'm speaking continuously, so that is happening almost in real time, right? So the that processing is also very fast. So imagine that how fast will it be when it even has a regional center to look at, right? These kinds of responses. So the body felt, or uh, however we were designed, it was felt that this reaction time is not sufficient. It might burn my finger till that point, till the time I realize that I have to remove the finger away, right? So it has to be even faster. So that is why this kind of a method was inculcated, wherein there is a reflex which is getting controlled not through the brain but through the spinal cord itself. So, over here, when we talk about a reflex action, like any other action that is generated in response to a stimulus, it has three kinds of components. One is a sensory signal, which will basically be something that the stimulus is generating and it is perceiving the stimulus. One is a motor signal that is carrying this, right? And one is a muscle signal. Whatever response is carried back through the motor signal, the execution is happening via these muscle signals, right? So that is what is happening over here. Yeah. So over here, for instance, you say that there was some kind of a sensory stimulus that was given, right? There was an input. First off, there could have been a slight input. For instance, I was a needle was going to prick my finger, right? It would have touched my finger also at some point. It might not have gone in as yet, but it might have touched my finger at some point, right? So that would have uh, produce some kind of a stimulus, some kind of a response, and that might have been a graded potential, right? So I am still not feeling anything. Like I am feeling the touch of that particular needle, but I am not feeling that I any pain or that I need to remove it, right? So that kind of a thing might be happening over here. Eventually, when multiple or a multitude of the signal comes in, there might be an integration of the signal wherein an action potential is generated and a receptor potential is also present. Then there will be, once an action potential is generated, it will be taken to the spinal cord, right? So there will be conduction of this action potential that will happen. And how it will happen is that this particular neuron, it might have its cell body in the spinal cord itself, right? So this is a single neuron, say, or a group, even if it is a group of neurons at certain distances that are taking whatever they are picking up over here to the spindle. How they are transmitting this is through neurotransmitter release, right? So that is what is happening. Eventually, the final one that is reaching the spinal cord, what it is doing is it is again releasing the neurotransmitter, and this neurotransmitter that it is releasing is being picked up by this motor neuron that has a cell body over here itself, right? What this motor neuron will do, again, it will receive a signal. It will, stimulate some kind of a response. This response will be in the form of an action potential, which will be conducted again to that point, right? So this will be conducted to this point again, right? And over here, although one single, input was being brought in, the per the function of this spinal cord neurons is that it will divide, because not only this muscle has to contract, even this muscle has to flex in order for that knee, for the knee to, or that portion to move away from the needle, right? So there are multiple muscles maybe that will be involved. So the key function of these neurons in this spinal cord is that it will delegate all those actions simultaneously, right? So the input is single, but the output that is it is sending is maybe to multiple different muscles, right? Because it will be a compound effect of those muscles, maybe contracting, flexing, relaxing, etc., that will help the finger move away from the needle, right? And then again, obviously, because it is sending the signal to the muscle, it will again receive the signal. There is some kind of an integration of that signal that will happen over here as well. And the conduction will lead to again either a contraction or a relaxation, which is going to be reflected in the behavior that the muscle is performing, right? So this is a sequence of signals which produces a reflex action. When we talk particularly about the sensory signal, right, what happens? So, a stretching of a muscle produces a receptor potential in the specialized receptor, that is the muscle spindle, right? The amplitude of the receptor potential, which is over here, is proportional to the intensity of the stretch of that receptor, right? This potential spreads passively to the integrative or trigger zone at the first node of Ranvier. So, till the first node of Ranvier, it is going passively. There is no action potential that has been generated as such. If the receptor potential is sufficiently large, it triggers an action potential and then propagates actively and without change along the axon to the axon terminal. At specialized sites in the terminal, the action potential leads to an output signal, the release of the chemical neurotransmitter. The transmitter then diffuses across the synaptic cleft between the axon terminal and target motor neuron that innervates the stretched muscle, and then it binds to the receptor molecule on the external membrane of the motor neuron. So it is going from this point from which the stimulus it is receiving to the motor neuron eventually, right? Thereafter, what is happening at this motor signal segment? This interaction is initiating a synaptic potential that spreads passively to the trigger zone of the motor neuron's axon, where it initiates an action potential that propagates actively to the terminal of the motor neuron's axon. The action potential releases a neurotransmitter where the axon terminal meets a muscle fiber, right? So that is the response that this motor signal is going to produce. But okay, but how is that going to be read by the muscle, right? So the neurotransmitter binds the receptors on the muscle fiber, triggering a synaptic potential in the muscle. If it is sufficiently large or if it is combined with signals from other motor neurons, the synaptic potential will generate an action potential in the muscle and cause contraction of the muscle fiber itself, right? So that is what will happen. Then we'll understand a little more about these spinal reflexes. There are multiple kinds of spinal reflexes that could happen, right? It could either be this polysynaptic pathways or monosynaptic pathways. We'll understand both of them, right? Yeah. Yeah. So first off, when we look at polysynaptic pathways, which is a flexion reflex, basically, you can see over here as well. You can see the gluteus and this other muscle. So this is the forward part of the thigh, this is the backward part of the thigh, right? So this is the extensor muscle and this is the flexor muscle over here, right? So what we are going to understand is the flexion reflex, right? Over here you can see that if there is a stimulation on one leg, right? So probably something hit you on this leg, there is a stimulation that is getting transferred from this leg, right, to the spinal cord. So it is getting transferred by this cutaneous afferent fiber from the nociceptor, right? That is A delta. What it is doing it is taking it to the cell bodies in this lateral horn region of the spinal cord, and where the cell bodies of these motor neurons are, right? What it is doing is it is further taking this information, and it is giving information to different kinds of muscle, right? So when, for instance, you have a response wherein a knee flex is happening, right? what will happen? Both your knees will not flex, right? Whichever knee is getting hit will only respond to that, right? So if both of them respond to that, then that will also be a problem. So the challenge over here is that it needs to flex this knee, but it also needs the other this leg to remain stable so that it can hold your weight, so that you don't tumble altogether, right? So that is how it does this, through combination of neurons over here. So there are activated motor neurons in both the legs that is happening for that to happen effectively. So polysynaptic pathways in spinal cord mediate flexion and cross extension reflexes. Once excitatory pathway activates the motor neurons that innervate ipsilateral flexor muscle over here, which withdraw the limb from the noxious stimuli. Another pathway simultaneously excites the motor neurons that innervate the contralateral extensor muscle over here, providing support during the withdrawing of the limb. Inhibitory interneurons ensure the motor neurons supplying antagonist muscle are inactive during the reflex response, right? So it has to make sure that this is not giving the response. So there are inhibitory interneurons that are playing that role, right? But this is getting the response. So that, and again, as I told you in the previous discussion also, that only this muscle contracting will not do anything. This muscle should also flex. That is one should contract, the other should relax. Then only that movement happens, right? Even in your arm, if you say, then if this is getting contracted, this is getting relaxed, right? If this is getting contracted, this is getting relaxed, then only that movement can happen, right? Especially in your limbs. Then we come to this monosynaptic pathway or the stretch reflex, right? Which is for the uh, single side, right? So monosynaptic pathway mediates stretch reflexes. Afferent axons from muscle spindles make excitatory connections on two set of motor neurons. One is the alpha motor neurons over here, and the other is the which innervate the same muscle or homonymous muscle from which they arise, and motor neurons that innervate synergistic muscle. So synergistic muscle is this one, right? This is the homonymous muscle, and this is the synergistic muscle, the muscle next to it, which is also going to help in the contraction. They also act through interneurons to inhibit the motor neurons that innervate the antagonist muscle. This is the antagonist muscle over here. When a muscle is stretched by a tap with a reflex hammer, the firing rate in the afferent fiber from the spindle increases. This basically leads to contraction of the same muscle and the synergist and relaxation of the antagonist, as seen over here. The reflex, therefore, tends to counteract the stretch, enhancing the spring-like properties of the muscle. The records on the right demonstrate over here, right? Sorry, here demonstrate the, oh, I have not kept that image of these records, so you can skip that part. Okay. Then we can briefly also talk about A.V. Hill, which was one of the scientists who was talked about. He was one of the people who even discovered cats first, right? So that is why also he's very important and he's also relevant. Also independently, not related to this neural work, but independently also he was a Nobel Prize laureate. So he also won the Nobel Prize in Physiology or Medicine in 1922. So as early as 1922 he won this. He was a prize share of 1/2, like it was divided into two people. The other was Otto, I don't recall the second name of that person, right? But you can go to the Nobel Prize archives and you can check that. His prize was for his discovery relating to the production of heat in the muscle. So he was, at the time he was working on these muscle fibers, contraction, relaxation, etc. What he had found, so this is a brief of why he won the Nobel Prize. So we know that in order to work, our muscles need energy, right? Which is released by chemical processes. So even neurotransmitter release, for instance, is a chemical process, right? During and ATP release also is a chemical process, right? During 1910s, Archibald Hill outlined these processes by studying muscles from frogs. In opposition to prevailing view that mechanical movement and chemical processes were parallel sequences, Hill was able to show through measurements of heat generated by mechanical processes that these were delayed in relation to the movements. The chemical consequence consists of a work phase which is not dependent on oxygen supply and a recovery phase when oxygen is required. So there is a hypoxic part and an oxygen dependent part of our even this uh, ATP production cycle, right, within the mitochondria, within the cytoplasm. So he was the conceptualizing person behind it. Then we'll also briefly discuss about Nansen, who was also mentioned by Sir. He was a neuroanatomist. He was a Norwegian scientist, actually, also the first important and internationally known Norwegian scientist who studied the brain, right? But along with that, he was also a Nobel Prize laureate, but he was not a Nobel Prize laureate in physiology or medicine. He was actually a Peace Prize laureate, and we'll come to why that happened, right? But this excerpt that I have picked is from FENS Forum 20, the 130th anniversary of FENS Forum, which is Federation of Neuroscience, Federation of European Neuroscience Society. So that the on the 130th anniversary, they basically recognized Nansen again, and what they wrote about him is written over here. He was at the forefront of research at his time. His doctoral thesis, The Structure and Combination of Histological Elements of the Central Nervous System, formulated for the first time the idea later known as neuron doctrine. Right? So we commonly say that Hall was the person who basically formulated neuron doctrine, but at least in this reference in by Fence, was said that he was actually the person who was the first known person who supported this neuron doctrine, right? What it said was that the nervous system consists of separate nerve cells called neurons rather than a continuum of tubes on which the subsequent, one second, on which a subsequent brain research is based. Nansen also was the first to state the significance of neuropil, which is also called as dotted substance or with the interlacing of a nervous fibril between the nerve cell bodies as the site of nervous activity and communication underlying consciousness and intelligence. Right. So let us go through his Nobel Prize record also. So this was Nansen, right? If you look at his Nobel Prize fact record, you can see that in again 1922, the same prize that A.V. Hale was awarded the Nobel Prize for Physiology or Medicine, he was awarded the Nobel Peace Prize. Um, and he was born in Norway, he died in Norway, he was a Norwegian all through his life. And the motivation, and he was the sole winner of this Nobel Prize, right? The sole motivation for his prize was for his leading role in the repatriation of prisoners of war in international relief work and as League of Nations High Commissioner for Refugees. So that was his main job. Sir mentioned about Nansen passport, right? So this is a Nansen passport, for instance. It is a copy of or a very early documented copy of one of the Nansen passports. So these Nansen passports was given to refugees at that time, or people who had no country or these kinds of things, right? So in 1922, the Norwegian Nansen became the first High Commissioner of Refugees appointed by League of Nations. After the First World War, he was in charge of the exchanges of around 4 lakh prisoners of war between uh, Russia, Germany, and former Austria-Hungary. Nansen also engaged in humanitarian relief work in 1921. Before that, during the severe famine of the Soviet Union, in his work on behalf of prisoners of war, starving people earned him the Peace Prize. He was a scientist, polar hero, political activist, and a diplomat as well. He got a PhD in zoology in 1888. So back in 1888, he had already earned his PhD. In the same year, he was the first to cross Greenland's inland ice. He subsequently failed to reach the North Pole, but became internationally famous nevertheless, right? So because he was also famous as an explorer, polar explorer. Nansen was a nationalist activist when Norway broke out of its Soviet Union with Sweden in 1905. After 1922, the League of Nations provided Nansen passports to stateless refugees to enable them to cross international borders. Nansen himself was made responsible for separating Greeks and Turks after the war between the two countries. In the last years of his life, he took up the Armenian cause as well, right? So, okay, so that was it for the lecture part of it. Now we are going to begin to understand this journey of how that electrical circuit is formed in the neuron, right? How whatever is happening within a neuron, we can compare it to an electrical circuit. What is the cause of driving of that circuit? Right, eventually we will build up to understanding that, and then we will again, as I told you, understand the Nernst equation, GHK equation, etc. Right. So when we talk about the driving forces, right? So these are two chambers, chamber A, chamber B. You can take them as a representation of the intracellular and extracellular side of the membrane, right? So you can say that chamber A is more positive, chamber B is more negative, right? Because it has more number of anions over here, it has more number of cations over here, so it is positive. It has more number of anions over here, so it is negative, right? But when you look at ions, right? What do you think will be the ion flow from chamber A to chamber B or B to A, right? So when we talk about the positively charged cations, that is potassium ions, you can see that there are 4 potassium ion molecules in chamber A, but there are around 7 potassium ion molecules in chamber B, right? So we know where, in what direction do the ions flow. It moves from its higher concentration to its lower concentration, right? So what will be the flow of ion over here? It will, the potassium ion will go from chamber B to chamber A, right? So concentration gradient for this potassium ion will be B to A in this direction, right? For the chloride ion, again, there is less number of chloride ion in chamber A and more number of chloride ions in chamber B. So again, in which direction will this flow happen? It will also happen from B to A. That is why over here we see that the concentration gradient is made in this direction, right? From B to A. But when we talk about the electrical gradient, right? So, uh, because inside is more negative, so the negative charge will probably try to flow outward, right? And because the outward is more positive, the positive charge will try to flow inward, right? So how will this inner membrane become positive? It will also be because of the flow of electrons, right, from one side to the other. And where will be the electrical gradient? It will be opposite, because there will be more flow of positive ions from outside to inside to basically neutralize this inner negative charge, right? So the electrical gradient in this case will be in the opposite direction of the concentration gradient. So these driving forces are basically what result in the value of the membrane potential, right? And we know what is the value of the membrane potential, roughly about minus 70 millivolt, right? So now look at it in actual context of the plasma membrane. So this is intracellular side of the plasma membrane, extracellular side of the plasma membrane. What it says is if we open the potassium channels and allow ions to flow outside, right? The energy that will be released per mole, this is based upon the Nernst equation we'll know later, is RT natural log of Ki by Ko. So where Ki is the concentration gradient inside and Ko is the concentration of this potassium outside, right? So if this ratio is more than 1, what it means is that ion ratio of ions is higher inside, right? And then the ratio is positive. But positive energy is being released when potassium ions are moving out, right? So that is the case of electrical concentration gradient. Now, when we talk about electrical gradient, right? So we can consider outside to be almost zero and inside to have a negative potential, suppose E volt, right? So energy released per mole for inward movement will be minus Z into E into F, right? Where Z is the charge of the ion that we are talking about, which will be positive +1 for the potassium ion. F is the Faraday constant and E is the membrane potential, right? So energy released per mole for outward movement will be opposite of this. It will be ZEF. So only the negative symbol will not be there, right? So because potassium ions are positively charged, they will be attracted toward the negative potential. That is inside the cell. Although the concentration gradient was driving the potassium ions from the inside to the outside, the electrical gradient will try to bring potassium ions inside the cell. Right? So that is what is going to happen. When we talk about equilibrium state, the net energy change is zero. So what does equilibrium mean? That there is no change of ions that is happening within this plasma membrane from either the inside to outside or outside to inside. No net change is happening, right? So that means it will be at equilibrium. So at this condition, ZEF plus the natural log of RT natural log of Ki by Ko will be zero, right? So for any ion X, over here K was this X, right? Instead of K, it could be Na also, it could be Cl also. For any ion X, the Nernst equation, which was derived by Walter Nernst in 1888, was Ex is equals to so the equilibrium that it requires for this particular ion X is equals to RT by ZF is equals to into natural log of XO by XI, right? So this was the equation that was derived. Out of that, we have three constants over here, which is no, so two constants, which is R, that is the Cass constant, and F, which is the Faraday constant. There is also absolute temperature, which we generally take as the room temperature, and the valence of electron, which will depend on which ion we are talking about, right? For instance, for calcium it will be positive 2, for chloride it will be -1, and concentration outside the cell and concentration inside the cell, right? So Ex is called the Nernst potential or the reversal potential or the equilibrium potential of the ion X, right? We can further cut it down because as I told you, our like temperature we are always considering as a standard room temperature, which is either 25 degrees Celsius or 298 Kelvin. So RT by F, because we have two constants R and F, and T also, if we always consider it as 25 degrees Celsius, we can always say that it is approximately RT by F is approximately 25 millivolt, right? We can calculate it and we can find out. So the Nernst equation, it automatically becomes Ex is equals to 58 millivolt by Z into log XO by XI, right? So if we try this Nernst equation in case of the squid giant taxon, wherein these recordings were first done, right, or accurate recordings were first done, what you can see is that in case of potassium, the concentration within the cytoplasm is 400 millimolar, but concentration in extracellular fluid is only 20 millimolar, right? When we talk about these values in the Nernst equation, we find that what is the equilibrium potential of potassium over here? It is minus 75 millivolt. Right, so at minus 75 millivolt, electrical gradient and chemical gradient for K plus ion will be balanced out. Right, now let us also understand what is this value for sodium and chloride, other important ions, right? Okay, we'll eventually come to that. So equilibrium potential for potassium is minus 75 millivolt. If membrane potential is equal to that value, then no net movement of potassium ion, because the electrical gradient and concentration gradient will eventually be balanced out, right? So what happens if the membrane potential goes below minus 75 millivolt? Right, what will happen? Electrical gradient will become stronger and it will attract K plus ions from outside, which will bring the membrane potential back towards this potential, right? So all every time, what will the exchange, even the electrical gradient or chemical gradient, what will it try to achieve? It will try to achieve this potential for that particular ion, right? That is the constant thing that keeps happening. So what will happen if it goes above minus 70 millivolts, say minus 60 if it goes to, then the electrical gradient will become weaker and the chemical gradient will push some of these towards outside, right? So that again it can bring it closer to minus 75 millivolt, right? Because it is positive, if it goes out, then the negative will again start increasing, right? So concentration gradient changes more slowly than the electrical gradient. Now that is a concept that we are aware of, right? Because ion transfer happens more slowly than the current transfer, right? So if you take this in a representation of a K plus channel without the concentration gradient, right? If you only consider an electrical gradient, so in absence of concentration gradient, equilibrium potential of potassium, for instance, is zero, right? So this is the extracellular side, cytoplasmic side. This is again a conductance of a single channel. This is a channel. The conductance of the single channel is equal to opposite of its resistance, right? So conductance is 1 by R. C is equals to 1 by R. It is opposite of resistance. So that is what is written over here. So by Ohm's law, you can say that I is equals to V by R, or it is equals to I is equals to gamma V, right? So slope is equals to gamma K. That is the conductance of this particular ion, right? In the presence of concentration gradient, however, the EK will not be equal to zero. There will be no ionic current, but V will be equal to EK. If V is not equal to EK, the current will flow through the channel, which will push V towards EK, right? So it will try to push this voltage towards again the equilibrium potential of potassium. So this will actually be the slope, but it will try to push it towards the equilibrium potential of this thing, potassium. So V above or below EK will result in potassium current with the same IV slope as below as before, right? So this will be the equation for that, and this is what we see. right? So this will be the potential difference that is created, and it will represent the potential equilibrium potential of EK. So if we have to represent it as a battery, this will is what it will look like. So this is a ion channel for potassium. This ion channel has certain conductance, right? After that conductance, it has to reach an equilibrium potential. So this is a potential difference. For that, we have introduced over here this voltage or potential difference component, right? So when we are talking about multiple K plus channels, there is not just one channel, right? There are multiple K plus channels that are in play or at play at a single point of time, right? When there are multiple potassium channels, then this conductance will be a result of the number of channels into the conductance of potassium in general. And this is what the circuit will look like, right? Sorry. Yeah. So, EK is not dependent on the number of channels, so EK is going to remain constant, right? So equilibrium potential for a particular ion is going to remain uh, that much only how many ever channels we have, right? If it is minus 75 millivolt for potassium, it is going to remain as minus 75 millivolt. If it is plus 55 millivolt for sodium, it is going to remain plus 55 millivolt only, right? If we talk about the circuit model, right? Till now, you were looking at this part of it. There is an extracellular side and an intracellular or a cytoplasmic side. There is a particular channel for a particular ion, which, and over here the conductance or resistance also, you can write it as resistance also. Over here, conductance of that channel is given, the potential of that channel is given. And when these components are present, there will also be a capacitance that will be present, right? So we'll come to that in a few minutes, right? So over here, what you should note is that different ions have different equilibrium potentials, right? So potassium, we saw that the equilibrium potential is minus 75. Sodium, it is plus 55, and chloride, it is minus 60, right? So because these are the major ions, right? There are others, there are calcium, there's magnesium, but these are the major ions in terms of how much of this ion is present. You can see that two of them are more negative and only one is more positive. So that is why the total equilibrium potential or resting membrane potential of the plasma membrane itself is more negative, right? So it is close to minus 70. It is basically an evening out of multiple of these ion channels, right? So if, for instance, ever different channels for sodium, potassium, and chloride, this is what the circuit would look like, right? So in this circuit, you can see different kinds of additions now, right? Wherein the potassium and chloride is more negative inside towards the cytoplasmic side, and Na is more positive inside, right? So it is a little inverted in this case, right? So E denotes only the magnitude of the Nernst potential over here, right? So the K plus ions would try to take the membrane potential close to minus 75 millivolt. Na would try to take it close to plus 55 millivolt, and chloride will try to take it close to minus 60 millivolt, right? So this is going to constantly keep happening. That is why that equilibrium is continuously changing, right? So there is an equilibrium that each of these need to maintain, and it is different from the other, right? So that is why it is continuously changing. So usually at rest, there are more potassium channels that are open than the sodium channels. So what happens is we can consider the case when only potassium channels are initially open, such as V is equals to EK, right? So over here, the membrane potential is going to be equal to potential for potassium ions, right? So then a few sodium channels also open up. Then what happened? The K plus channels are first only open, right? So the net driving force is potassium, right? and the chemical driving forces towards outside and electrical driving forces towards inside, as you can see by this colored arrows denoted over here, right? Then a little bit of sodium, few of sodium channels also start opening up, right? So once this happens, once a few more sodium channels also start opening up, what will happen is that this sodium will also play a part, right? So the net driving forces now will be towards inside for the sodium channel, because the chemical as well as the electrical forces are towards inside, right? So the net driving force now changes towards inside, as you can see via this arrow, right? Then there is a steady state again that is achieved, wherein what happens is that these active channels are no longer open, but the sodium is driving sodium ions inside. So both electric and chemical currents. In potassium, the chemical current is stronger, which is driving it outside, and the electrical current is smaller, which is driving it inside, right? So there is a net driving force wherein more of sodium is getting driven inside and less of potassium is taken outside. But the net result of this current is somewhere around here, which basically is the representation for a steady state current, wherein the membrane potential is at almost the resting membrane potential or the equilibrium, right? So at steady state or rest, Vm between the equilibrium potential of potassium and sodium, right? So this is the equilibrium potential of potassium. This is the equilibrium potential of sodium. Although it is not like midway between them, but it is somewhere between them, right? So this is the steady state potential, right? So at steady state, the potassium ions are continuously trying to still reach minus 70. So K plus is still moving out. The sodium ions are still continuously trying to reach plus 55. So it is moving in. If the sodium and potassium current balance out, right, then membrane potential stays at this equilibrium, in this steady state potential, right? So if we use equivalent currents to determine a circuit to determine the resting membrane potential, this is what it will look like, right? Similar to what we have seen ago earlier. So there has not been much addition till now. But now we are going to understand these components also eventually. So at steady state, there is no change in V, but the net current flowing out of the membrane should be zero, right? So thus V minus Na into the conductance of Na, V minus Ek into conductance of K, and V minus ECl into conductance of Cl is equals to zero. So this is the equation that we get. And this comes to be around minus 69 millivolt in case of the squid giant axon, right? So what you can see from here is that this value is not simply an average of the three, right? So if you take out an average of all the conductances, that minus 75 plus 55 minus 60, it is not an average value, right? Then it would have been somewhere in the middle, but it is a weighted average, right? It is weighted by the conductance. So how much it is getting conducted is weighing in on this average. So it is a weighted average. That is why it is coming closer to the potassium, right? Because the conductance for potassium is more, right? So if potassium conductance of potassium is higher, then the final voltage of will be closer to potassium's equilibrium potential, which we can see over here. We know that the potential conductance of potassium is the highest in the resting state. Therefore, it is not surprising that this value comes closer to the equilibrium potential of the membrane, right? So equilibrium potential of membrane is minus 69 and that of potassium alone is minus 75, right? So it is closer to that. Then, if you look at a simplified representation of a resting membrane, then again, this is a simplified version of the same equation. These passive channels together are known as leakage channels, right? Because nothing is invoking them to become, transport electrons. It is not an active transport, it is a passive transport. So it is known as leakage channels. Then, if we assume that potassium, sodium, and chloride currents balance out, the membrane potential stays at steady state potential. But this can't go on indefinitely, right? Concentration gradients will deplete because constantly these individual channels are trying to put bring it to their equilibrium potential and not to the steady state potential. The concentration gradients are maintained in parallel by active transporters such as NAK pump, which have non-zero net currents, right? So this is an active transport mechanism of maintaining these concentration gradients, which we have seen multiple times, right? So this is the symbol for this NAK pump, right? So you can add this to the circuit as well to make it more acceptable. So you can see that the passive transport is faster, but the active transport via the pump, it consumes energy and it is slower relatively, right? The net current through the ion channels is not zero as we assumed in the last slide. So we come to this GHK equation or Goldman equation as it is known in short. This is that the resting membrane potential is equals to RT by F, natural log of all these ions, right? The potassium, the potential of potassium ions outside versus inside. Outside, again, we will take potassium and sodium and chloride and any other ion that is there. Upon their potential difference inside, that is for potassium, sodium, and chloride, right? So this is the GHK equation. Right. So again, Vm is the resting membrane potential or the steady state. Px is the permeability of the ion. X denotes either potassium, sodium, or chloride in this case. And for squid giant taxon at rest, this ratio, the pK by PNA by PCl, that is the permeability of these ions at rest, is 1 is to 0.04 is to 0.45, right? So maximum it is for potassium, then for chloride, then for sodium, which also we have mentioned briefly in previous lecture, right? So this is the GHK equation. Now we can consider the case of glial cells, right? In case of glial cells such as astrocytes, microglia, the but this this yeah, permeability of this ion, potassium is high, right? But it is almost negligible for sodium and calcium. So we can remove these for sodium and calcium, and it basically will become a Nernst equation if it is a for a particular ion. So we can also consider Nernst equation to be a special case of the GHK equation itself, right? Then what is the cause of action potentials, right? The cause of action potentials is ion channels, these leakage ion channels, right? So how does this happen? We'll understand that. Just give me a moment. Yeah. So these ion channels or the leakage channels are the ones that are actually causing these action potentials to occur, right? So what are the properties of these ion channels if we want to understand this, right? So they conduct ions at high speeds. The conduction of ions across the cell membrane at very rapid rates, up to 100 million ions per second. So it is that high, right? That rapid. They recognize and select specific ions. So there will be a specific chloride ion channel, specific potassium ion channel, specific sodium ion channel. So for instance, over here you can understand that potassium ion channel is 100 times more permeable to potassium than sodium. And even sodium ion channel is 15 times more permeable to sodium than potassium, right? So, some channels open and close in response to specific electrical, chemical, or mechanical signals also. Some are open all the time, some open and close with respect to these changes that are occurring around them, right? So, how do these ion channels become selective? Let us understand that quickly, right? Before moving on to that circuit part again. So, for instance, we'll tell you the crystal radius of potassium as well as sodium, right? So, potassium is actually bigger in size, 0.133 nanometer, and sodium is only 0.095 nanometer, right? So, ideally, what I would have thought is that how are they becoming selective? So if it was like a sieve, right, that only a particular size of the ion can pass through a particular channel, it would have been good. But for instance, you can say that a smaller ion channel will let this thing pass, but it will not let this particular thing pass, right? Potassium will not be able to pass, right? But a bigger ion channel, which has a pore size equivalent to that of potassium, will let potassium pass, and it should also let sodium pass, right? Because sodium is smaller than potassium. So if it has a bigger pore size, then this can also pass. But on the contrary, what have we studied in the previous slide? That the specificity for the potassium ion channel is much higher for potassium than for sodium. The sodium can sometimes let the potassium also pass, but because it is only 15 times more permeable for sodium than potassium, but potassium channel is 100 times more permeable for potassium than for sodium, right? So how does this happen, right? So yeah, is there some chemical moiety that helps is binding to it, which is selecting sodium and not potassium, or and potassium or not sodium, right? Like that, is it happening or what is happening, right? So the earlier thing that I explained, that there will be a particular sieve size maybe which will let one ion pass and not the other. That is true surprisingly for potassium, right? How? Because although it is bigger in size, what happens is because of because these ions are in an aqueous medium, right? The sodium ion, because it is very small, what happens is the aqueous medium contains many water molecules, which are seen over here. The oxygen atom is more electronegative than the hydrogen atom, right? It tends to attract the negative charge towards it, creating a partial negative charge on oxygen and partial positive charge on the hydrogen ions. Out of sodium or potassium, ions tend to attract these partially negative oxygen atoms because of their positive charge, right? And what happens? As a result, sodium is never present in isolation. Rather, it is always surrounded by a bunch of water molecules, right? And because uh, uh, the radius of this electronegative charge is actually higher in case of sodium than potassium, right? This results in an increase in effective diameter of the whole molecule. These bunch of water molecules are known as hydration shell, and ion with the hydration shell has much larger radius than the ion alone, right? So over here you can see the potassium also has a hydration shell, and sodium also has a hydration shell. But the hydration shell of sodium is much larger, which makes its effective size larger than that of potassium ions. The smaller sodium ion, because of more localized charge, attracts more water molecules and has a larger hydration shell than potassium ions over here. Thus, Na behaves as if it is larger than potassium, right? So now that logic will work, right? So this explains how a channel could select potassium ions over sodium ions based on the diameter of the pore, right? So because this has a lower diameter, it can pass, but sodium will have a more diameter, so it will get stuck. It will not be able to pass, right? So this we understood how the potassium ion is selective for potassium and not sodium. But how do sodium channels become specific for sodium, right? That also has to occur somehow. So how does that happen? So the ion channels are not rigid tubes, but they have proteins that can interact with the surrounding molecules. This was a second theory, right? So the ion channel for sodium has a selectivity filter. Now, how does the selectivity filter work? Right? So this is what the inside of the ion channel of for sodium looks like. So what happens is the proteins are made up of amino acids that have various kinds of side chains. When ions are passing, they interact with the side chains of amino acids within this channel. Right? What it does is the ion channels have a narrow region in the middle known as selectivity filter. In these narrow regions, hydration shells cannot pass as intact molecules. So they break down, right? The hydration, so the water molecules which are stripped off like this, so it is stripped off, so only sodium or potassium ion will remain, right? The removal is an energetically unfavorable reaction, but it can take place because sodium or potassium atom can be stabilized by interaction with negatively charged amino acids, right? So because the side chains are helping, that is why even this energetically unfavorable reaction is able to occur in this particular channel. After this, the ion can pass through it through and it can regain the hydration shell like this case, and interaction will depend on the exact positioning of these various amino acids, right? So somewhere along these lengths, these amino acids are present, for instance over here, right? In sodium atom, the positioning is such that the sodium can be fully stabilized, so it is optimized for size of sodium atom, but for potassium, it will not be that true, right? Then we come to the gated ion channels, right? What is gating? Let us understand. So some ion channels have two or more conformational states that are stable, right? One could be an open state and a closed state, right? The transition of a channel between these states is called gating. Such channels can be called as gated ion channels, right? So not every channel is open all the time. The ones that are open all the time are known as leakage channels, right? But some are either open or closed, right? So because they're switching between these states, it is called as gated ion channels. So a gating is applied, right? So how does these conformation or do these conformational changes occur? It could either be a conformational change in one region, for instance, over here, the pores particularly that is helping, that is open. If it is open, it is helping a channel pass, but if it is closed at this point, it will not help a particular ion pass through, right? Or the general structure itself could change, right? So either the channel would be like this when it is open, but it will be like this when it is shut. The entire channel is shut, right? Or there could be some kind of a blocking particle that is present, right? For instance, over here you can see in the C terminal, there is a particle when it is opened, it is removed, but it is physically blocks the channel when it is closed, right? So there are several type of stimuli that control the opening and closing of these ion channels, right? These could be ligands. These channels are known as ligand-gated channels. So if a ligand comes and binds to the site, a particular receptor or a particular site in this channel, then it brings it to an open conformation. It could be a voltage gated channel. The change of membrane potential could cause the opening of the channel, or it could be a stretch or pressure gated channel as well, as is seen in this third example, right? So under the influence of these regulators, channels enter one of the three following states. Either it is closed and activatable, which is its resting phase, or it is open, wherein it is active, or it is closed and it is non-activatable, which is its refractory phase, right? So this is important for understanding that action potential part, why it cannot travel in the other direction once it has started or begun its course, right? So because of the presence of these closed and non-activatable channels or refractory channels at that point, right? So then we again come to understanding this equivalent circuit, right, wherein there is an ionic current and a capacitive current that takes place. In case when any recording is done for these cell membranes, the membrane current can be divided into either capacitive or ionic current. The ionic current is further divided into leakage currents, which happen through due to passive transport of these ions, voltage gated sodium or voltage gated potassium currents, right? So in voltage clamp also the capacitive current is zero except when the holding or command voltage is changed. The leakage current is constant for a given command voltage, right? So when we talk about again a voltage clamp recording, you can see that small depolarization that is moving towards that zero will basically cause some change in the voltage itself, but it may or may not cause a change in the conductance, right? So IM over here indicates the compensatory current injected into the cell, right, to survive that particular change that is occurring. But once we consider the voltage gated conductances, then the picture becomes more complicated. How? It is basically not easy to figure out whether the observed current is due to the voltage gated sodium channels or through the voltage gated potassium channels. This is where some chemicals become very useful, right? So now we will understand how we understood in the first place why this current is happening. Is it because of the sodium channels that it is happening, or it is because of the potassium channel it is happening? So if we have to find this out, right, in another situation, if you take it, for instance, I am saying that whenever I light a candle and I turn on this white light in my room, I get a headache, right? So now these are two conditions, right? So one could think that either the lighting of the candle could give me a headache, or the white light presence could give me a headache, right? But it could be both as well, right? How will I filter that out, right? Once I will only turn on the white light and not light the candle. Once I will light the candle and not turn on the white light, and I will observe what is the effect that is happening, right? So similar kind of a study was done over here also, right? That is where some chemicals were useful, right? To turn off one kind of a channel so that particularly the other kind of a channel could be studied. So one of these chemicals was tetrodotoxin or TTX. It is a poison that is found in pufferfish and some other animals as well. So pufferfish, you would know that whenever a prey approaches this fish, it basically swells up like this, so that it will be difficult for the prey to eat or prey on this fish, right? So because it is so puffed up. So and even if it does eat it, it has this toxin, tetrodotoxin, and what this toxin does is it basically blocks the voltage gated sodium channel. So it will directly cause conduction issues in that uh, you know in that animal that is trying to prey on it. So it is more poisonous than potassium cyanide, 1,000 times more poisonous than potassium cyanide, right? Because it is directly going to intervene with all the chemical transmissions, right? And then there is no living after that. Then there are many natural poisons which function by blocking some aspect of the neural activity, therefore lead to muscle dysfunction or cardiac arrest or respiratory problems, right? So it is generally because of these conduction issues that they do, right? So, uh, okay. So when we talk about an action potential, this is current from a large depolarization, this kind of a current we are getting, right? So a large capacitive current, initially there is an inward current and then there is an outward current that happens, right? As the leakage conductance is constant, leakage current can be easily computed and subtracted, right? So that we can take care of. But there is not just leakage conductance that is happening, right? There are also active conductances for sodium and potassium, for instance, that is happening. So we can separate voltage gated sodium and potassium currents if we block specific channels, right? So how we do this? To block the sodium channels, we use tetrodotoxin, and to block the voltage gated potassium channels, we use either TEA, that is tetraethyl ammonium, or 4-aminopyridine 4-AP, right? What happens is we will sequentially look at these experiments. So sequentially, when these channels are blocked, then what happens, right? So generally, we see this kind of a current. Total current is like this, right? When the sodium or potassium channels were blocked and the current was measured, what was seen was that in the presence of TEA, when potassium currents are dropped, basically this kind of a peak is seen. So this is a peak only for the sodium, right? Because potassium is blocked over here. And when in the presence of tetrodotoxin it is seen, then only the peak for potassium is seen because sodium is blocked. So it looks like this. So the total current that we see over here is a summated response somehow of this potassium and sodium, right? But you can see that this part of the peak resembles very much with the sodium, right? That will come in handy later, right? So if we repeat the same experiment for different values of the command voltage and measure the voltage gated currents, then this is what it looks like, right? So when only potassium ions we are measuring, then it looks something like this, right? So the net driving force at plus 75, everything is outward, right? Electrical, chemical, gradient. So net driving force is also outward, right? At minus 50, because it is closer to the equilibrium potential of potassium, then the chemical is still outward because ions are more in number inside and less in number outside, but electrical now has inverted because the inside is more electronegative, right? So the net driving force is much less at this point, right? Similar for sodium, if you see initially at plus 75, because it is higher than its own potential, which is plus 55, the chemical will be inside, but the electrical force will be outside. At its own steady state, it is equal, so the net driving is zero. But as it begins to polarize, right, then what happens? The net driving force shifts inwards, right? Because both the chemical and electrical will eventually be inward. So these are the conductances of sodium and potassium, which can be measured from the currents of sodium and potassium. This is the formula for the same, right? And this is what it looks like. So both the conductances change with depolarization. Hence, the name voltage gated is given over here. Note that there is difference in speed of change. That is, sodium is faster in this case, right? Because there are more number of sodium channels also that are present, right? So this conductance of sodium and potassium are functions of uh, the potential. That is, Hodgkin and Huxley determine the exact dependence of this, right? So if you depolarize a neuron and don't clamp the voltage, right, or V, then V changes will change depending on the conductance of sodium and potassium. And this study also won them the Nobel Prize in 1963. This is what they have solved, right? So if you see the action potential that we get, it is partly due to the conductance of sodium and potassium. So over here also you can see that because the peak is attained very quickly, right? And then there is this refractory period that occurs, right? So this is suppose minus 70. This is depolarization, this is repolarization, and because it is going a little more negative than the actual resting membrane potential, it is basically known as hyperpolarization, right? So how does this happen? You can see that this action potential peak is very similar to the sodium conductance peak, right? So because sodium ions open very quickly, right? Sodium ion channels open very quickly. So according to the Hodgkin-Huxley model, an action potential involves the following sequence of events. A depolarization of the membrane causes sodium channels to open rapidly, which increases the conductance of sodium. This current causes after further depolarization, thereby opening more sodium channels, resulting in more inward current. The depolarization then limits the duration of action potential in two ways. It gradually inactivates the sodium channels that had opened earlier, and it also with some delay, potassium channels also start opening. So it starts declining. Potassium channels will open, then again the equilibrium will fall more towards the potassium, right? So consequently, inward sodium current reduces and outward potassium current increases. That tends to hyperpolarize the membrane, right? So why does that hyperpolarization this part occur? Because at this time you can see the sodium is almost closed, but the potassium is still open, right? So it will take it further down from resting membrane potential closer to its own potential of potassium, right? So it will further hyperpolarize. But by the time the sodium also finally start opening, it will start to recover to the resting membrane potential itself. Right, so that is the cause. So membrane potential goes below the point from where it had actually started, which is known as after hyperpolarization, right? Because it is happening after the depolarization cycle has happened. So over here you can see membrane potential is determined by the balance between these different ions. The sodium ions try to take the membrane potential towards their equilibrium potential, which is towards its positive, uh, uh, while the potassium ions try to take the membrane potential towards their equilibrium, towards minus 75 in this case. During the action potential, the sodium conductance has increased, but then it also inactivated, right? So earlier it increases very quickly, but then it starts deactivating also, or getting inactivated, while potassium conductance increases a little later, but it also remains active for longer, right? It does not deactivate. So potassium ions are still open at this point. Therefore, potassium keep on going out and the membrane keeps becoming more negative. Because we have additional potassium conductance here, the membrane potential will go towards the equilibrium potential of potassium ion, which is towards minus 75. But it will not go below this point, below the equilibrium potential of potassium at least, right? So, as voltage gated potassium channels gradually close, the membrane potential can eventually return to the baseline. So, when we talk about the action potential threshold and amplitude, you can see that the threshold is around this point, right? If this threshold is crossed, then the action potential amplitude that we get is the same, right? It will not be more or less depending on more or less stimulus, right? So, subthreshold stimulus will not produce any action potential, but it will give an equivalent graded response, right? Threshold stimulus will produce an action potential, but even if there is a supra threshold stimulus, that is stimulus is much stronger than the threshold what is required to cross the threshold, the action potential will not be that much stronger. There will just be an action potential, right? So this is the supra maximal response that is obtained in case of an action potential. So there are two factors that begin to act. The voltage gated sodium channels open up, which try to further increase the potential difference, and the voltage, sorry, and the leakage channels which try to decrease the voltage. So the action potential size depends on the voltage gated channels and does not depend on the stimulus size by itself, right? So if sodium channels are inactivated for some time after the action potential, which is what is actually happening, then what happens is because the sodium ion are closed, right? The resting channel over here is closed and it cannot be activated till some time. So it is in that stage closed and inactivatable, right? So this is the first case. Then there is after a particular time it is activated. It is over here in the first case it is closed and activatable. Once it is activated, it opens up. Sodium passes inside, but then it is eventually again closed and inactivated for some time, right? Then it requires some time and some repolarization to occur for it to become activatable again. This is what the refractory period of sodium looks like, right? So there is a delay in this period of activating of the sodium channel, right? This is why it is not activatable for some time. So if you try to activate it with the same threshold very quickly, it will not produce even at the same amplitude, it will not produce that much threshold. It will not cross the threshold. But if the timing is at least to some few milliseconds apart, it will be able to touch the threshold again eventually, right? Because more sodium channels will become activated, reactivated by this time, right? So this refractory period happens. There is a current clamp that we can use to measure this. The refractory period is amount of time which is in a few milliseconds, after which a spike, after a spike in which it is impossible, which is a absolute refractory period. This one absolute refractory period, it is impossible to again stimulated, or the relative refractory period wherein it is difficult to get another spike, right? In absolute, you can definitely not get a spike. In relative, if the threshold is very much, then maybe you can get a spike. The reason is inactivation of sodium channels or closing of the potassium channels as well, right? So this is the end of the slideshow for now. Uh, and that is it. We'll be discussing the electric circuit part in more detail maybe in the next lecture, right? So I have taught what is ahead of that, that is the time constant and the length constant. But while I was revisiting it, I thought it might not have been very clear because we have not explained what exactly is the capacitance, what exactly changes are happening. So I would want to revisit that again. Let us see like how much of the next week's course material we still have to discuss. Accordingly, we can plan that, right? So for now, that will be the end of the discussion from my side. If you have anything to say, please go ahead. Else, I'll just end the recording and thank you for attending.